All right, so let's get going with our keynote presentation. To kick off this journey of our considerations and insights for scaling non-viral gene delivery, we have Heinrich Haas, who is the Vice President of RNA Formulation and Drug Delivery at BioNTech. So welcome, Heinrich, and thank you for joining us today. Take it away.
Okay, yeah, thank you very much for this kind of introduction. It's a pleasure to me to share some of our research in this beautiful and very high-level symposium. Let me just start right away. So I would like to share some short thoughts with you on RNA nanomedicines, where I give you short overview over development aspects, starting from basic research until clinical level, and have organized the presentation in the sense that I first give you some introduction into RNA therapeutics. I will talk a little bit about general R&D, RNA and formulation development. I will show some slides on clinical product development, and then with that I will conclude and hopefully be in time. So RNA therapeutics, as probably you all know, the RNA nanomedicines comprise messenger RNA as a therapeutic molecule, which needs to be introduced into a cell where the RNA expresses a certain protein of interest, which can be an antibody, an antigen, or any other therapeutic protein where there is a certain need by the patient. Now, for development of RNA therapeutics, you need to take into consideration several aspects. So you need to make sure that RNA synthesis and optimization can be done. You need a formulation for the RNA to allow administration to the patient. The formulation mostly needs as well to deliver the cargo to the target site. And in certain aspects, you also need to realize a personalized manufacturing of the products in case of personalized cancer therapy, for example. The computer is slow. As mentioned, there are different therapeutic options for which RNA can be applied in our organization. We use many of them for practical development, including cancer immunotherapies, infectious diseases, obviously, and also other indications where protein replacement or other aspects of RNA therapeutics play a role. I would like to focus a little bit on cancer immunotherapies, which is one of our key interests in pharmaceutical development. And in that case, we do something similar to a vaccination, where however we use cancer tumor antigens as targets. So tumor cells are characterized by certain tumor-associated antigens, which can be recognized by the immune system, and similar to an immune response to a foreign antigen, pathogen. Also, cancer cells can then be attacked by our immune system. So in order to do such a vaccination or to stimulate therapeutic response against that, what we do is we take an RNA which codes for a tumor-associated antigen, which is then transferred into dendritic cells, which are having a key function in tumor therapy. And in this dendritic cells, the RNA is translated into the antigen, and this stimulates a whole cascade of immune responses, both specific towards the tumor antigen, but also a systemic immune response, which helps for the cancer therapy. Now, I would come to some aspects on how to develop such formulations to allow for that. And first of all, of course, you do need a therapeutic RNA. The RNA is an active pharmaceutical ingredient. And here you see an overview over typical structural elements of an RNA. In the middle, there is the coding region, which comprises the information required to synthesize the protein. But there are also other very important elements like the 5' and 3' UTR regions. There's this cap, which has for activity, and there's this poly A tail. All of them need to be optimized individually, and all of them synergistically help to make a real pharmaceutically applicable molecule out of the messenger RNA. Here you see something on synthesis. So the good thing is that RNA can be manufactured in cell-free processes using enzymes, where you start with the DNA plasmid, which is then linearized. Then you translate that into the messenger RNA using the nucleotide, and then it's purified. And in the end, you have the pure RNA molecule for further processing. The RNA needs to be formulated for delivery, and these formulations, they are required to do several things similar to classical drug delivery for cancer therapy with small molecules. And here, potentially, the challenges are even a bit higher. So functions are, for example, that RNA needs to be protected from degradation in circulation. You have to control the serum interactions in the manner you want them. The RNA needs to be delivered to the target site where you want to control biodistribution. The formulation must enable or even improve cellular uptake at the target site, and it also needs to allow for translation of the RNA and release of the coded protein, if required, into circulation. So there's a cascade of functions which need to be fulfilled by the formulation. And as you all know, the classical way to do this is to form nanoparticles out of the RNA and the vehicle, which can be lipid or polymer based. And this nanoparticle is then manufactured as a pharmaceutical product. And after injection, which can be parenteral, intravenous, or intramuscular, the RNA is supposed to be delivered to the site of activity. Here we see some typical molecules which serve as vehicles. We have lipids, which are used for the lipid-based nanoparticles, NLPs and others. There are cationic or cationic ionizable lipids, which are required to bind the RNA to the lipid matrix. There are helper lipids, where here you see also typical candidates which are mixed together according to the formulation you want to arrive at. And maybe another branch of delivery systems, which is not so much in the focus at the time being, but which is important as well, are polymers, for example, polyethylenimine or other polymers, which are as well positively charged in order to allow binding and compaction of the RNA for delivery. Here, I would just briefly mention some important aspects and paradigms which are considered to be related to activity of lipid-based delivery systems. And one of those is that the phase state of the lipids plays an important role for the capacity to enable transfection. And here you see cartoons which depict lamellar phases as present in liposomes, or hexagonal phases, inverse hexagonal or classical hexagonal phases, and certain lipids are known to be able to undergo transitions between these different phases, and those are considered to be particularly helpful as lipids for transfection. Another important concept is the optimization of the pKa value of ionizable lipids. And here you see two examples from literature where different ionized moieties are shown, and you can see that the pKa value changes depending on the chemical nature of this functionalized moieties. And this is extracted from a paper where using siRNA. It has been found out that a certain pKa value is best for best transfection efficacies, and this is generally accepted nowadays in the community, and I will come back to that in the later presentation. How do you make formulations? So simply spoken, you just take two different or several different starting phases, one comprising the RNA, one comprising the vehicle, which can be liposomes or lipid solution or polymers. You mix them together with buffers in an appropriate manner. You end up into nanoparticles, which you then characterize by their physical-chemical characteristics, and you evaluate the activity in cell culture or animal models, and then you can get this correlation between particle characteristics and biology, and you can use that as a basis for optimization of your systems. Here you see some two typical examples of lipid-based delivery systems. On the left hand, you see lipoplexes, which are typically formed with liposomes as the starting phase, so the lipids are here present in the lamellar phase in water. They are mixed together with RNA, and they form this lamellar-like lipoplex stacks, which, for example, can be used in cancer therapy for targeting APCs. And here you see the LMPs, which are a bit more complex in composition, and which, in contrary to the lipoplexes in that case, comprise an ionizable lipid. They comprise typically cholesterol. They comprise a stealthy moiety, which is required for particle engineering. And these are those which are frequently used for the COVID vaccines, but also for many other applications. So these systems are somehow similar, as they consist of very similar components, but still they are a little bit different as I said, in composition and also in other properties. And one aspect where these systems are different is the internal structure. This can be demonstrated, for example, by small angle X-ray scattering. Here I show you how this works in principle. You take your sample and irradiate it with an X-ray beam, and you just collect the scattered light, which you then integrate circularly, and you come up with scattering profiles where you get the intensity as a function of the angle, and this is here the momentum transfer, which is corresponding to the angle. And one parameter you can derive from such patterns is, if it's present, the type of Bragg peak, which is characterized by a peak position, a peak width, and a peak area. And all of them give you information on the internal organization of your particle. Here, for example, what you see is scattering patterns for lipoplex system and LMP system. And again, you can outline some similarities and some differences. So the lipoplexes are characterized by a relatively sharp Bragg peak, which is narrow and well-defined, with an otherwise unstructured profile to lower Q. While the LMPs there don't have such a sharp peak, but still some kind of peak-like pattern, which is way broader, and the modulations are visible also to a higher Q. And these are related to internal characteristics, which allows for specifying the system, and also which can provide a basis for optimizing them once you would like to reveal certain parameters. I will come back to that a little bit later with concrete examples. Another difference between LMPs and lipoplexes is the fact that in the LMPs frequently so-called ionizable lipids are used, and in lipoplexes permanently charged cationic lipids. And this could be also something where you look at a little bit more carefully in order to reveal similarities and differences. And for example, here you see sister molecules which are identical in structure except one motif at the head group, which makes them in one case permanently charged. In the case of DOTMA, here with a T, and the DOTMA, there's this methyl group missing. This makes it ionizable, and you can switch the charge as a function of pH from cationic to neutral. Same for this other pair of lipids, DOTAP, the cationic one, and DODAP, the ionizable one. And coming now to some structure elements, this is a little bit more complex summary of X-ray scattering patterns which have been generated from systems comprising ionizable and permanently charged lipids together with RNA and alone. And here you can reveal important structural features. This is a measurement where we changed the pH of the system from acidic to neutral. And if you go to a classical cationic RNA lipoplex, you see that these patterns are all the same, irrespective of the pH. We again have our nice peaks, which gives us information on despacing and organization, but the pattern doesn't change so much. If we're going to the other end, this is a system comprising ionizable lipid together with RNA. And here actually you see that as a function of pH, the peak position changes. And this change means that the packing changes, so the repeat distance of this regularly ordered arrays. And in this manner, you can get direct structural information on the internal organization of the systems as a function of pH. And if you plot these, for example, in that case, the despacing, the repeat distance as a function of pH, you can get classical curves, which, for example, the cationic system shows no change. And the ones which comprise an ionizable lipid and RNA, they show a sigmoidal curve where there is a plateau region in the extremes, but there is a range where the spacing changes. And this can be then kind of translated into a phenomenological pKa value similar to other methods. And such data allow you to get direct information, as I said, on the packing density and other packing characteristics at low and high pH, which allow to predict certain occurrences, for example, in circulation and after cellular uptake on endosomal processing and release. And interestingly, you can get very, very accurate information from such system. Here you see, for example, from the previous data, the pKa value, which was determined as a function of the NP ratio, so of the fraction of RNA inside the model membranes. And what you could see is that the effective pKa changes as a function of NP value, and by that manner you can kind of in a tailored fashion control and predict accurately pKa values with a very high resolution and on purpose. And this can be something which is helpful in addition to chemical synthesis, as we saw in some slides before, where people synthesize different lipids and then investigated pKa values of LMPs with certain chemical compositions, where you always need to synthesize a kind of a lipid library. Here we can tailor these on the basis of composition aspects and other aspects of the lipoplex system, which allows you to much easier and more accurate adjusts these parameters. I would now come back to a little bit simpler system, which is being composed by mixing the RNA with the vehicle, again, liposomes in different ratios, and show you some slides on these lipoplex systems, which can be assembled with a different mixing ratio, and where you can use either an excess of liposomes or RNA. And as has been shown already some time ago, some years ago, if you do such an experiment, you mix RNA and liposomes in different ratios, you can find regions where you make colloidally stable particles, either with an excess of negative or positive charge. And these particles can then be again characterized and investigated. And as has been demonstrated, and which is now in clinical practice, by doing so, you can control the preferential delivery and expression profile of RNA in vivo. This is experiments using luciferase as a reporter gene, where the lipoplexes were injected intravenously. And as we see, if we have lipoplexes where you have an excess of liposomes, there's a very high transfection efficacy in the lung, while if you have an excess of RNA, there is a very high and selective expression in the spleen, just by changing that physicochemical parameter. This can be then kind of semi-quantitatively analyzed, and on this basis, you may define development candidates for pharmaceutical products in case you need to deliver to some pharmaceutically active component to the lung or to the liver, which is both being in practice for different therapeutic interventions. So if you have identified such a formulation, this is not the end, this is kind of the beginning of all the development work, because then you have to refine all these parameters and to justify them. And for that, I always recommend people to look at the FDA liposome guidance, which is, as the name implies, originally written for liposomes. And the FDA instructs us to how to characterize the system and what data to generate. This is an extract of this liposome guidance where all kinds of parameters are listed, which should be known and reported. And these are basically, to a large extent, as well applicable to lipoplex and LNP systems and RNA delivery systems in general, as in both cases, in all cases. We talk about nanoparticles, where the internal structure and organization plays an important role for activity. Now, if you do upscaling, typically here you see an example for liposome manufacturing. You start with something adopted for lab scale experiments, like this extruders here, which is not so suitable for working under GMP. So you may change your process on making this liposome, this colloid, and this is then to be transferred into a GMP environment where you manufacture it in larger scale according to regulatory requirements. To do so, to allow for that, you need to control, determine, and justify all process parameters much more accurate and with much more care than this was done for the formulation experiments. And in principle, all critical quality attributes need to be justified and determined and controlled for allowing to have the GMP manufacturing established. And just going back to our previous example of this NP ratio variation, now you can use X-ray scattering to justify, for example, the right NP ratio to come up with a certain formulation and to define the criteria under which manufacturing can be done, which can be accepted, and those where you leave the secure range of composition and manufacturing conditions. And this is for an example where such an experiment has been done. Again, X-ray scattering measurements have been used to study lipoplexes assembled at different NP ratios. This is in excess of RNA, and this is in excess of liposomes. And you can see that, again, we have our Bragg peak, which in this range where you have a negative excess, the peak characteristics doesn't change so much, but it increases, meaning that more or less you increase the amount of your lipoplexes, but the lipoplexes stay somehow the same over the whole range of negative excess, which is very good in terms of robustness of the manufacturing protocol. While on the contrary, if you have a positive charge excess, the pattern changes, the peak is still there, but it becomes broader and it shifts, and it indicates that here there is a transition or composition, maybe a superposition of different patterns which superimpose to the final X-ray diagram. And in the end, such measurements, so this was just one example for one measurement, for one parameter. This allows you, for example, to derive, I called it a phase diagram, where you identify certain ranges where product characteristics, for example, stay in a wide range, virtually invariant. They are maintained, and others, in other ranges, the characteristics change, so they are unstable. And then you could also outline regimes where you may have, let's say, different patterns which superimpose to each other, and by changing the ratio, you can identify the conditions you want to have the optimized characteristics of your product. So in a nutshell, doing many of such measurements for many of such parameters, you can define all process conditions you do need to manufacture a pharmaceutical product. This includes concentrations, pH values, presence of ions and buffers, compositions of lipids, again, buffers for the liposome phase, mixing conditions, microfluidic architectures. You also can specify compositions of them, the ready-made lipoplex nanoparticles or LNP nanoparticles, and these then are being filled and according to the necessities stored in the liquid, frozen or lifeless state according to the requirements of your pharmaceutical product. So with that, I would like to come to the end and maybe summarize some points I wanted to share with you in this presentation. So these CMC aspects I shared with you are extremely important to successfully develop complex nanoparticles as those consisting of RNA and other complex molecules. So RNA as a drug substance, it's labile, it's highly charged, it's complex as mentioned, and it makes pharmaceutical development also a greater challenge than in case of small molecules. For example, formulation is of key importance for such products, because formulation may tremendously affect the product qualities, and technologies from the nanoparticle world are important tools in order to define the formulations and to develop them towards practical, clinically developable products. A thorough understanding of these coherencies of structure and function as derived from advanced characterization, such as X-ray scattering, but many others, can help to perform formulation development, to save lots of efforts which would otherwise be done by screening techniques. And this also helps you to define the critical quality attributes which you do need to specify your product. And all those are important important criteria for successful translation of the RNA into clinical development. And with that, at the end, I would like to thank the formulation team and of course Uwe Stein, who made all of this possible. Many of the measurements I shared with you have been done in cooperation with Peter Langut's group at the University of Mainz and the Pharmaceutical Department. Some of our systems we obtained from Matthias Balz's group, who's now in the Leiden University in the Netherlands. Our lipids are partially from cooperation with Dan Pierce group in Tel Aviv University. And the X-ray measurements to a large extent have been done at the EMBL outstation at Daisy Hamburg, together with Martin Schröer, Clement Blanchet, and Dimitri Svaragun at that station. With that, I'm really at the end. Thanks again very much for listening, and I'm happy to take questions.